Good morning, everyone. You can grab a seat. It's my pleasure this morning to introduce Dr. Zhu. He is a new faculty member in the Department of Pathology. He received his MD and PhD at Soochow University and came to the U.S., did a postdoctoral fellowship at Wash U in St. Louis, and then went back into clinical work and did a pathology residency, and then went on to do a heart and lung specialization at the Armed Services Hospital. He's run an NIH-funded lab now for well over a decade. He has almost 100 publications to his name and over 50 invited talks. He was recruited here starting in the summer to head our head and neck pathology group. Those of you who are in the cardiomyopathy and heart transplant group have probably already started interacting with him. But I thought it was a good idea, since he will likely be working with many of you and hopefully coming to our M&Ms to do the pathology review, that we have him here to speak. So thank you very much.
Yeah, thanks to Rob's introduction. Also, I'm very glad to be here to give a presentation of my research. My lab basically right now is focused on the regulation of cardiac sodium channel. So this probably you guys know more than I do. Basically, the electrocardiogram is determined by electrical properties of individual cardiomyocytes. As you can see, the P wave is representative of atrial depolarization. QI represents ventricular depolarization. QT represents ventricular depolarization and repolarization. PR interval basically is the time from the signal transfer from the atria to the ventricle, starting of the ventricular depolarization. So as you can see, this is typical ventricular myocytes activation action potential, which has been divided to 4 phases. Zero phase is depolarization phase. Is determined by rapidly inward and outward inwardly sodium currents. Then phase 1 basically is determined by transient outward fast potassium current. Then phase 2 is determined by L-type calcium current. Then you say type 3 is determined inward rectified potassium currents. Then these 2 currents bring the action potential down to the resting membrane potential, which is determined by inward rectified potassium currents. Actually, during my KO8 training, I was doing lots of research on the transient outward potassium current, which is called like ITO. After my KO8 training, I have moved on to the sodium channel regulation. So as just to summarize what I did in my KO8 training, basically we determined the transient outward current is determined by one particular potassium alpha subunit Kv4.3, which can be phosphorylated by multiple like kinase, such as P90ISK, which is elevated in multiple cardiac disease, such as cardiac diabetes, cardiac heart failures, etc. So this enzyme activation then directly phosphorylates the C terminus of this alpha subunit. As you can see, potassium alpha subunit is kind of different from sodium alpha subunit. I would address that later. So each potassium channel consists of 4 alpha subunits. As you can see, the P loop between S5, S6 basically contribute to forming the potassium selected channels. So as you can see, after this channel is phosphorylated by P90ISK, then peak currents downregulated. The duration of APD duration is prolonged, then QT interval is prolonged. basically we consider P90K activation in the diseased hearts is important in the regulation of potassium current. Most likely is a trigger to cardiac arrhythmia as well. So after my KOA training, actually moved on to the research in cardiac sodium channel regulation. As I pointed out before, sodium channel topology is different from potassium channels. As you can see, this is one molecule actually consists of 4 domains. Each domain, like potassium sodium channel alpha subunit. As you can see, 6 domains, transmembrane domains of the main domain 1, for example. Then you have loop between S5 and S6, which contributes to form sodium selector channels. So also this channel can be modulated by multiple elements, including cytoskeleton protein and some beta subunits, beta 1 to beta 4. So why we are kind of very interested in sodium currents? When I was apply the grants during my even the KO8 application, so they already criticized me. So why did you choose the ITO? Because ITO is not important as this current in the small animals. I think after a couple of years later, actually, KV 4.3, which underlines the transient out of faster transient out of potassium current, actually is related to Brugada syndrome. So I think also it's important. By that time, actually, I switched more to the sodium current regulation. So why sodium current is very important? As you know, this current is universally present in the small animals and the humans. So also, this channel mutations have been identified to lung QT syndrome, such as type 3 lung QT syndrome. Several mechanisms which can underline this kind of different type of syndromes. Lung QT type 3 mainly is due to the mutations of the sodium channel, which mutation actually causes gain function of the sodium current, as you can see, which basically make the prolonged of the APD duration. So in this cardiomyocyte, it's easy to have EAD or activity. Also, as you can see, Brugada syndrome is due to the mutation of sodium channel, which basically make the channel decrease activity. So as you can see, you see the decreased sodium channel. You have typical patterns, ECG morphology of Brugada syndrome. Also, you have some persistent cardiac conductance disorders, which is due to sodium channel mutation as well. So that's from the genetic studies, then linking to the human disease. So how about the mouse models? So if a homozygous deletion of these channels actually cause the leader, so they actually, some groups choose a hemizygous deletion of some sodium channel. As you can see, this is basically SC5A genes, which determines the NA1.5 proteins, which is mainly underlines the cardiac sodium channel. Y type, you see the very nice ECG consisting P wave, QIs, etc. But some mice with hemizygous deletion of sodium channel show you kind of ventricular tachycardia. Also, from this animal, is suggestive of these genes is very important for cardiac physiology as well. So as you are cardiologists and may know much better than I do. So after the ischemic heart disease, the patients are mostly within 24 hours, even within 48 hours, majority of patients have died of the ventricular tachycardia or either ventricular fibrillation. So actually, what exact mechanism is underlying this arrhythmia? Basically, our lab's long-term goal is want to understand the mechanisms which underline this kind of arrhythmias process. In my laboratory, basically, we can do multiple animal models, such as myocardial infarction, reperfuse injury, to try to find the mimics of a human myocardial infarction. As you can see, this is a sham procedure of the mouse heart. That's basically ligation or reperfusion cause injury. Then you can have myocardial infarction. based on the echo data, we can find the cardiac function significant decrease after myocardial infarction. Also, in my laboratory, we can do EP studies as you can do in the patients. So basically, after myocardial infarction in mouse model, as you can see, the mouse is very difficult to develop cardiac arrhythmias even after myocardial infarction. Actually, we can use the stimulation like EP studies to determine this is after myocardial infarction. This mice can easy to be generate the cardiac arrhythmia as a ventricular tachycardia and atrial fibrillation. All this basically by S1, S2, even S3, the stimulation to cause the mice to develop the cardiac arrhythmias after myocardial infarction. So as other proteins like sodium channel protein, basically is modulated at the different levels. Actually, you can see at the transcription levels, usually this gene's promoters can be modulated by multiple transcription factors. In this reviews, actually make a reference to our working. Actually, FOXO1 is a transcription factor. Actually, can suppress this gene expression. So that this genes after transcription at the different levels actually modulate this function of sodium channels. So today, basically, I'm going to focus on my talk, which consists of two portions. Which first one, I'm going to focus on the FOXO1 transcription factors regulating the sodium channel. Next portion, talk depends on time, how much I can talk. That we are going to talk about Wnt beta-catenin signaling pathway, which is also suppressor signaling pathway on the sodium channel activities. So why do we have kind of such interest in these two signal pathway? So my long-term goal is want to understand the mechanisms of myocardial infarction patients with cardiac arrhythmias. First, we try to understand several transcription factors activities changed after myocardial infarction. So that, as you can see, this is a human heart failure hearts. As you can see, FOXO1 is significantly translocated to the nuclear side. Also, we did the myocardial infarction to measure the sodium channel messenger RNA protein levels from the border zone of myocardial infarction. Actually, as you can see, both messenger RNA and protein levels of sodium channels proteins are significantly decreased. That's potentially linking between these transcription factors and sodium channel expression. Also, we found in the human heart failure, beta-catenin and signaling TCF4 signaling pathway also activated. So this immunohistochemistry staining show this is beta-catenin is translocated to the nuclear side in the ischemic heart disease of human tissue. So both actually beta-catenin and FOXO1 signal pathway kind of most likely is involving the ischemic heart disease, or could be regulating the sodium channel expression. So FOXO basically has different isoform, FOXO1, 2, 3, and FOXO6. FOXO1, 2, 3 basically is they are highly expressed in myocardium. Also, FOXO1 is kind of more rich in myocardium. So this basically general little demonstrator of the function domains of this FOXO. So all transcription factors which always have a DNA binding domain. This FOXO had a DNA binding domain. So today this molecule has a different function domains as well: nuclear localization sequence, nuclear exporting sequence, as well as a transaction domain. So as you probably know, this FOXO can have multiple functions, particularly in insulin growth factors signaling pathway. This FOXO activity is most dependent on the AKT activation. After phosphorylation of FOXO, actually, this is a molecule is translocalized the nucleoside, then play multiple functions in cell death and endogen homeostasis, etc. So when we do studies of the transcription factors regulating the some targeted genes, we usually use alignment among different alignment promoter areas from the genes, such as like this is from promoters from the sodium channel. Then align them, make alignment among the different species. So we found potentially FOXO1 binding sites so that we can do chip assay. That's basically a routine we're doing chip assay. If this is a FOXO1, can be recruited in the promoter area. We use one is cell line, HL1 cell line. It's a cardiomyocyte cell line. Also, we do chip assay in the myocardium. We found basically FOXO1 is recruited in the promoter region of these channels. So next question we'll usually do in the in vitro experiment to answer if this is a proteins is very important for the promoter activities. We do like luciferase assay. As you can see, we clone the genes of the promoter 2.3 kb in front of the luciferase genes. So as you can see, this is basically FOXO1 is dose dependent. This promoter activity is dose dependent of FOXO1. When we increase the FOXO1 expression, actually the promoter activity significantly decreased. So we do mutation analysis. We mutate this particular FOXO1 binding site. As you can see, this promoter activity reverse also is increased by these two mutants. So we also overexpress this kind of proteins in the HL1 cells. As you know, HL1 cells is original from the mouse HL cells, actually motorized to the cell line. So as you can see, when we overexpress this FOXO1, actually it's mutants. This mutants basically change the amino acid serine to the alanine, make this FOXO1 is activated. As you can see, this overexpressed FOXO1, you see the protein of the sodium channel expression at both protein levels and messenger RNA levels are decreased. Also, at that time, we cannot explain why. So when we overexpress this activated form FOXO1, we saw FOXO1 also increased the expression. That's kind of endogenous FOXO1. Well, we actually looked the literature. So actually, just they published the paper in JBC. They found actually FOXO1 has the promoter of the activity. Promoter activity can be regulated by itself, FOXO1. So that's exactly explain this why the FOXO1 can increase FOXO1 expression itself. So certainly we are, from our experience, we want to know the cardiac sodium channel activity is regulated by these genes. We basically overexpress this is activated form FOXO1, as you can see, significantly downregulate the sodium channels. So today we are going to do like silence of these genes experiments. We always use like a multiple experiments to determine this genes. This genes plays a very important role regulating the sodium channel from positive or negative ways. As you can see, we use the microRNA silence this gene's expression. So basically, you can see this sodium channel messenger RNA and protein levels are both increased by silence of the FOXO1 expression. So as you know, like a hydrogen peroxide is a significant increase the myocardial, ischemic myocardium. So we want to know this is a H2O2 functions in the regulation of the sodium channel through could be through the FOXO pathway. So we already know the H2O2 suppresses the sodium channel cardiac activity. So that basically we want to know this is H2O2 could be affect the translocation of FOXO1. H2O2 has been reported at lower concentration can activate the ATP. ATP then phosphorates the FOXO1. After FOXO1 phosphorylation, basically this FOXO1 is nuclear localized. So then we want to know how the H2O2 regulates the sodium channel expression. So as you can see, when we add the lower concentration, 25 millimolar, this concentration basically does not cause the apoptosis of the HL1 cells. So, but this significant decrease the sodium channel expression. So the next question we want to know, the HL2 is also kind of important in the regulation of the FOXO1 suppressing the sodium channel expression. Basically, we use this as a silence of the FOXO1 also in the presence of the hydrogen peroxide. As you can see, basically this is a downregulation of FOXO1, basically prevent the H2O2 effects on the sodium channel expression. So we did like a bunch of studies in the vitro, basically how do you convince people say, oh, this FOXO1 is very important in vivo. So by that time, we actually did like get the FOXO1 looks P mice, then cross with Cray image, basically Cray mice, as we basically use a cardiac-specific adult tamoxifen-induced deletion of the FOXO1 in the heart to see the cardiac sodium channel is activated or downregulated. As you can see, this is a deletion of the FOXO1 sodium channel is robust, basically significant increase of the deletion in both messenger levels and protein levels. Surprisingly, actually, we found like a beta-3, which is beta subunits, which can regulate the alpha subunits of sodium channel expression, also robust increase by deletion of FOXO1. So this basic experiments told us this is a FOXO1. It is a suppressor of sodium channel expression in the in vivo system. So the question always comes to here, say, do you think this is a downregulation of sodium channel? Is due to the cardiac function change. As you can see, we performed the echo studies. After the deletion of FOXO1, about 1 month, we performed the echo study. As you can see, EF and FS actually was not changed. Heart body weight ratio is not changed. Also, cardiomyocytes, the size was not changed, indicating FOXO1 deletion did not affect the cardiac function, even cardiac hypertrophy. So we move on the EP studies. So we isolate the cardiomyocytes, then to do action potential recording. As you can see, after the deletion of the FOXO1, basically we marks of the phase 0 depolarization of action potential is significantly increased, which basically leads to the shortening of the QRS. As you know, the QRS duration is mainly determined by sodium channel activity. So this also consists in the finding of the FOXO1 deletion. So then we are basically recording the sodium currents from this FOXO1 deletion cardiomyocytes. As you can see, after deletion, probably sodium channel activity is increased about 1/3. The kinetics of the sodium channels basically were not changed much. So from this portion of talk, we can conclude that FOXO1 is basically a suppressor of the sodium channel expression. Deletion of the FOXO1 actually is increased sodium channel activity. FOXO1 also involved in the modulation of sodium channel, presumably in the ischemic heart disease, because H2O2 is a significant increase in ischemic heart disease. So next portion of my talk, I'm going to talk about Wnt beta-catenin involving the sodium channel pathway. So briefly talking here, this is a cartoon showing how beta-catenin activation involving the genes regulation. As you can see, when the Wnt signaling is activated, the complex, basically including APC axin 2, GSK3 kinase beta-3. So basically, this is it's a complex. It's very important to determine that GSK3 kinase activity. When this pathway is activated, basically GSK3 is inactivated. Phosphorylation of the beta-catenin is inhibited. After basically inhibition of phosphorylation beta-catenin, most beta-catenin is less degradation, then move to the nucleoside, play important roles in the targeting genes. As you know, beta-catenin is a co-transcription factor, so that's interact with TCF4 transcription factor. The TCF4 usually binds to the promoter region, then forming complex with beta-catenin to target gene expression. So we do the same thing. We made alignment of the promoter of this sodium channel genes from the different species. We found several spots of the potentially TCF4 binding sites. So we do the same thing, do in vitro studies of luciferase acid, then overexpression of the beta-catenin. As you can see, this is a sodium channel promoter activity significantly depressed by the beta-catenin overexpression. As you know, lithium chloride is the inhibitor of the GSK3 beta. So after the inhibition of this kinase, kind of probably increase the beta-catenin, then suppress this channel's activity. So indicating this is beta-catenin may be involved in the regulation of sodium channel expression. So we did the immunoprecipitation to confirm beta-catenin can interact of the TCF4 in the ventricular myocardium. We did the immunoprecipitation, as you can see. Beta-catenin can be poured by beta-catenin antibody. Then you do the TCF4 antibody staining. As you can see, the TCF4 can be poured out by antibody of beta-catenin, indicating beta-catenin can interact with TCF4 in the myocardium. We do the same thing of the CHIP assay in myocardial, in HL1 cardiomyocytes and ventricular myocardium. As you can see, both beta-catenin and the TCF4 can be recruited in this sodium channels promoter region, indicating both TCF4 and beta-catenin can form in complex to regulate the promoter activity of the sodium channel genes. So when we always do some positive experiment, negative experiments, as this experiment, we use the deletion of N-terminus of beta-catenin because in this area, which contains the GSK3 beta subunit phosphorylation amino acid. When we delete it, basically, actually, the beta-catenin is less degraded by GSK kinase, beta-3 kinase. As you can see, after the overexpress this activation form beta-catenin robustly, as you can see, protein levels by Western blot, sodium channel, both expression at protein and messenger levels actually decreased. So we often do the electrophysiology on this kind of HL1 myocytes. When we overexpress, when we treat this myocyte with a lithium chloride, as I said, which is an inhibitor of the GSK beta-3, particularly as somebody reported before, beta-catenin is increased by this GSK3 beta kinase inhibitor. So after this inhibition, as you can see, the sodium channel is significantly decreased. This kind of consistent with decreased expression of the protein levels of the sodium channel. So certainly from another aspect, how about the GSK3 beta, let's say, inhibit these channels, basically you can see this is beta-catenin significant downregulation, then sodium channels also following the actually change. So we want to know also H2O2's effects on this sodium channel could through the pathway. Before one group, I think, reported that H2O2 can affect the beta-catenin transnuclear localization. We repeat this experiment. As you can see, we add the H2O2, beta-catenin is translocalized to nucleoside. TCL4 is still stay in the nucleoside. So yeah, when we submit the prior data to the one journal, actually, I think particularly free radical biological medicine, this journal requires lots and lots of kind of data. So ask us how about time dependent, dose dependent. Actually, we came back. So we did lots of studies about the dose dependent and time culture, like culture time windows to see if this is also suppress the channels by dose dependent. As you can see, when we increase the concentration of the H2O2, basically the gradient decrease of the sodium channel expression. Also with the time culture in the H2O2 kind of the decrease gradiently sodium channel expression by this approach. So that we use the kind of siRNA knock down the beta-catenin also in the presence of H2O2 to see this H2O2 effects through this beta-catenin silence. As you can see, when we add the siRNA targeting the beta-catenin, actually increase this sodium channel expression. Then when we in presence of H2O2, as you can see, H2O2 basically effects is prevented by beta-catenin silence. So indicating H2O2 also kind of affect the sodium channels activity through the beta-catenin signaling pathway. So we actually got the animal models from UCLA, Dr. Arjin lab. So as you can see, exon 3 plus exon 3 deletion. Basically, beta-catenin exon 3 encoding the peptide, which consists of the amino acids of the phosphorylation site by GSK3 beta. When we delete this exon 3, so basically this is beta-catenin is activated. Majority of the proteins is nuclear localized. So as you can see, this is beta-catenin with exon 3 deletion, the significant robust expressed by Western blot. Then sodium channel expression significant decrease by this robust expression of the activated form of the beta-catenin. So as you can, this is just a quantitative data of the protein messenger RNA levels to see sodium channel. It is a decrease in the heterozygous deletion and the homozygous deletion of the exon 3 of the beta-catenin. So other subunits were not changed by this approach. So the question is, how about the currents? You saw lots of proteins degradation, but how about the sodium channel activity? We do the patch clamp recording the sodium currents. As you can see, exon homozygous deletion, the sodium current actually was significant decrease. Only one question we could not solve, could not answer during our presentation in the AHA. Multiple actually people come to say, why is here reverse potential is changing? So usually reverse potential change is due to actual channel itself change, not by some kind of modulation, not just by like a phosphorylation, etc. But we cannot figure out. So that's probably next step. We need to solve that problems. So we also analyze this kinetics after the exon 3 deletion, activation, and inactivation of these channels. Kinetics and were not changed by these deletions. Very interesting findings, actually, the recovery, that means the channels inactivated, then after recovery, do you see the recovery time is much faster in the deletion of the sodium channel? Presumably, this is a deletion, activation of the beta-catenin may cause some protein expression, finally affect the sodium channel kinetics. We are going to presume that to do more experiment to see the targeting genes presumably affect the sodium channel expression. So we also isolate the cardiomyocytes to do the action potential recordings. As you can see, the activation, the depolarizing phase of the zero of the action potential actually significantly decrease in the Vmax rate. So also this is consistent with the sodium channel decrease by the exon 3 deletion of the beta-catenin. So when we talk about cardiac arrhythmias in the mouse model, yeah, it is very difficult to see phenotype of arrhythmia. We always use some kind of induce approach to see if this mice can be induced to generate cardiac arrhythmias. So as you know, this is we often use this kind of freakinide. It's like the IC, the sodium channel blocker. So as you can see in the Y type of the mice, when you add the freakinide, so basically they induce the PV block PR interval prolongation. You do see atrioventricular little kind of blocking. You say P wave actually missing the QRS. But when you can see about 60% of this mice with exon 3 deletion, they can induce the ventricular tachycardia. So this kind of model actually raises the possibility this mouse model may be representative of Brugada syndrome, because the patient with Brugada syndrome usually has no cardiac arrhythmias. Only in certain situations, some like additional factors, they can induce a patient, this Brugada patient, to generate a VT. Also, we also further analyze the data, for example, QRS, what's going on. So in the baseline, we found that the QRS is prolonged after activation of beta-catenin. So then after 1 minute after administration of this medicine, you can see this QRS further prolongation. But after 5 minutes, 15 minutes, actually QRS prolongation, they are kind of equal. But most likely, this is maybe after the injection of this medicine, basically beta-catenin block is saturated. So as I summarize data, very interesting actually from this aspect of the experiments. In the 60 to 70% of mice of exon 3 deletion, so basically show that arrhythmia VT induced by this medicine. Also, we found this kind of cardiac function was not by deletion of the beta-3 deletion. So certainly we use another model. We talk about the beta-catenin and the TCF4 interaction. We basically deletion like TCF4 in the adult heart. Then to see this can be blocked by beta-catenin infection. So as you can, deletion of the TCF4 sodium channel was not changing much, but actually reversed by activation of beta-catenin, as you can see. Because of time windows, somebody already waved, so I have to quickly to explain. So this is after basically TCF deletion, sodium channel activity was not changed, but in the presence of TCF4 deletion, then activation of beta-catenin kind of reversed a little bit by TCF4 deletion. So that's actually last piece of data we found. In this TCF4 knockout mice, or presence TCF4 and exon 3 deletion mice, all these mice cannot be induced to generate cardiac arrhythmias by flecainide. Only the group exon 3 deletion, probably 70% of mice to develop cardiac VT. So this summarizes the signaling pathway. I already talked about activated Wnt pathway, basically beta-catenin activation. So we propose beta-catenin interact both TCF4 and FOXO1. We are currently doing the FOXO1, also can interact with beta-catenin regular sodium channel. We also found another, actually activate a MES1, actually is increase the sodium channel expression. We are working this MES1 studies. Also in my lab, we found some like chromatin remodeling, which also affected the sodium channel expression. Yeah, basically, I just keep that jump to the acknowledge slide. Actually, multiple PIs, I got their help. They provide agents, they provide animal models. From my lab, our lab pretty small. It's difficult to generate say some like Crelox P animal models. We usually adapt the people's models in the different fields. The vascular area, brain area, we cross this mice with our Crelox, particularly in the cardiomyocyte, to see the phenotype regulation of our channels. Thank you for your attention.
That's going on simultaneously. Where do they cross off?
So actually, beta-catenin can interact with FOXO. So that's we found also another lab found in kind of different signal pathway. We propose that most likely beta-catenin can actually form in complex with TCF4 and FOXO. So right now we are trying to cross the beta-catenin activation mouse model with the FOXO1 deletion model to see any kind of little prevention of the prevent the FOXO beta-catenin activation or not.
Have you looked at the sort of other aspects of the QRS regulation?
So yeah, that's exactly like you review my grants. Actually, yeah, sorry. Yeah, they talk about it because always when you talk about the transcription factors, actually always multiple targets, right? Why you say this is your target, your sodium channel? So yeah, that's why we kind of try to address if this is a regulation directly or indirectly. We always say this is a FOXO1 kind of overexpression or decreased expression. Particularly in this model, we did not see the cardiac function change. We look at the histology, we did not see cardiac structure changes. But certainly, we cannot 100% exclude that another indirect pathway affect the sodium channel activity. Yeah, thank you. I feel like, yeah, thanks.
Thank you very much.
I need to like.